Thank you. It's a pleasure to be back. And I would also like to reiterate something that maybe Olga didn't quite say, but should have, that this course is a lot of effort from her whole group. And I think they deserve a thank you from the community and also from NIH to do this, to write the grant, get the money, organize this. It's a huge amount of work to do this for 2 weeks. So congratulations. Okay, so this is a presentation that I think tries to bring together some of the things you learned and put it in a biological context. And the topic I chose here as an overriding topic is the reproducibility of research results. So I'll first mention why I bring up this topic, and then how what you learned fits into that field. I think it is a very complicated issue, but also one that is extremely important from scientific, but also science, political, and societal effort, especially in the time now where scientific research results and scientific facts and evidence are drawn into question by many. And of course, the easiest way to discredit science is to say, well, the other guy did something similar and got a completely different result, so it's basically the results were wrong. So I'd like to comment a little bit about that in the context of what you learned. So this is an outline. I would like to discuss why we need to be able to generate reproducible research results, very briefly for political reasons, but then probably more importantly, for scientific reasons. Then I will touch on 3 studies that were done in using this type of DIA techniques that you learned now, and I would like to answer the questions: if 2 or more people have their same proteomic data, can they generate the same results? So that's basically directly addressing the issue that you now addressed in these 2 weeks. How do we extract results out of data? Can we do this credibly and reproducibly? Then I would like to pose the question: if 2 or more people have the same sample, can they generate the same proteomic results using these DIA techniques? And then if 2 or more people do the same experiment using a cell line, do they get the same result? Presumably, if they use the right software tools, the right settings. So these are 3 points, and in combination they would indicate if the answer is yes, this would mean we would be able to use these methods to do reproducible research results, which would be a nice thing to achieve. Okay, so first I will make a few comments about why the issue even comes up. So this started some years ago when companies, particularly pharma companies, got nervous because they found that oftentimes they took something like a drug target that had been developed by or published by some academic research laboratory, and then they tried to implement that in their company and they couldn't. So there's a couple of companies have spent a sizable effort to investigate this issue in a systematic manner. So I think the first of these studies was from Amgen, and they assigned a large number of scientists from internal science to reproduce results from 53 articles in cancer research. And these were not just research studies that were published somewhere in a journal no one reads. These were in reputable journals. They could reproduce about 10%. Bayer is also a company. They examined a larger number, 67 target validation projects, and also in oncology, they could reproduce about 21%, and so on. So this then means that it is difficult for a scientist, even a very qualified scientist, to take something that's in the literature, port it to their own laboratory, and do the same experiment and come up with the same results. This is, of course, not good. And then this leads to a statement like this. Is from This is from a blog which says the NIH in the US government gives about $30 billion a year through NIH funding, which is mainly distributed in grants. If 10% reproducibility rate means that 90% of this money, 28 billion of 31, would be wasted. So this is, of course, not good. Then how are the taxpayers supposed to respond to the scientists applied for more research funding given these numbers? Would you give more money, of your own money, to someone who delivered such a result? This is a text from a blog that I copied out here. So this is clearly an issue that is in people's mind. And of course, there is also a somewhat science policy danger that if this gets to very spread out in this opinion to politicians who are funding research, especially science-adverse politicians, that they would say, well, we basically waste a ton of money, let's use it for something else. So, Nature has done a good job, Nature Publishing Group, to try to bring some order in this field or in this issue, because it is not really useful to say, well, results are not reproducible. This may well be, but we need if you discover a problem in science, you try to figure out why is it a problem, and you try to fix it. And so Nature has produced a number of papers and blogs and articles where they try to break down this reproducibility issue into various levels. For instance, the experimental design, reagents, drugs, cells, data acquisition, statistics, validations, and so on. And obviously some of these things here you now learned in this course. So proper statistical procedures, to some extent different modes of data acquisition, and I'll get back to that in the field of proteomics. This is just to say that they establish kind of a menu of topics which we can break down the overall problem and start to investigate why we may have a problem and where it could be resolved and where it could not be resolved.
So,
yes?
My quick response is, I just feel 90% of startups fail. We should stop investing in startups.
It's not public money that we invest in, right?
What?
It's not necessarily public money that we invest in.
It's not public money, but I think very few people would also say that we shouldn't be investing in startups.
Yes, of course, startup is a slightly different situation because you try to do something that really has never been done, and you project into the future that this might work out. And scientifically, this may be very sound, but it may not be developed into business. And here, of course, we would assume that if there is things are not reproducible, something is wrong with the experiment or the results. But yeah, I mean, it's a similar situation in startups. In that many things don't work out. So then Nature again did in two years ago or so, 3 now, they did a survey among scientists. They surveyed 1,500 scientists and asked, is there a reproducibility crisis? So these are not random people from the street, these are actual scientists. And about 90% of those said, yes, there's a significant crisis or a slight crisis. So this is clearly very, very well-noted issue in the scientific community. So that was, to summarize this thing, is why is reproducibility important? Because it is the result of reproducibility of results is a bedrock requirement of experimental science. This is what you expect. If you understand the system and you do something with that system, you should get twice or many times the same results, whether you do it in Boston or in Melbourne or in Zurich, it should be the same answer. If it's not, there's something which we don't understand. Poor reproducibility means, or is construed by many as poor competency, that people don't know what they're doing, or worse, that they cheat. And I think this is the most dangerous part for this political arena, that some might say, well, the scientists don't really know what they're doing, or they are all cheaters, and they just make stuff up to get citations. And this, of course, is very damaging. So I think it's important to think about the issue and try to figure out why things fail, if they in fact fail. So now I would like also to make some comments about scientific reasons why we would need reproducible research results, especially in this field of proteomics. And for that I use an example of a study that we did, and this is continuing. And I think it illustrates the changing attitude or changing perception of how biological research is carried out. Many of you who are biologists have trained in as students, maybe as master or PhD students. We would learn to do a controlled experiment. And what that means basically is you take a mouse or a yeast cell or whatever, which is the object of your study, and you try to keep all variables constant. For instance, you use the same strain of mouse every time you do the experiment, exactly the same age, same time of day, it's fed or it's fasted, same temperature. Sometimes even microbes are controlled in cages. Some mice are even grown without any microbes at all. So that is attempts to basically control every conceivable parameter. And then you would introduce one change. For instance, add a drug to the mouse, or delete the gene, or mutate the gene, and then see what happens. And then you would do a measurement, and you will observe something, and generally, this is now the dangerous part, you generalize the finding, and you would write a paper and say, if I delete this particular gene or add this particular drug, in the species mouse, this effect has been observed. So, and I think this is where a lot of things go wrong in this generalization. One should always write down this effect of this drug or deletion of a gene or whatever was measured in this mouse, given these as a function of these variables. And they're of course never or hardly ever described. So then that leads to certainly indication that people trying to do the same experiment may not do the same experiment, because a mouse is not a mouse. This is, we have to realize that especially genetic variability is an inherent property of any species. Species only survive in evolutionary time if there is variability. So if a species faces a threat, let's say a new virus, it depends on the fact that some may die, but not all will die. If we were all monoclonal and genetically identical in a species, the species would have died out a long time ago. So genetic variability is an essential element of biology. It cannot be controlled. It is there, but we try to control it in experimental biology. So there has been, of course, a lot of attempts been done, for instance, by the NSF, to systematically take strains of mice or flies or other species and map out their genome. And what is clearly observed, exactly as in a human population, that mice are different genetically and phenotypically, exactly as humans are different. And of course, one consequence of this genetic variability is at the center of the very big field of precision or personalized medicine, where physicians, for instance in Mass General, observe that patients who have the same disease phenotype react maybe different to treatment. So there is no reference mouse, there is no reference state, all results are conditional, and that I think is important to recognize. I would like to illustrate this now, how we can use this not as a problem, but as a source of learning new biology. That we would try to use the genomic variability that has been created through evolution in a species or across species, that we learn things about biology which we could not learn if we took just one strain and controlled all the variables. So I'd like to make this in an example which from project we've been pursuing for quite a while. So here we deliberately try to use mice which are not the same. So they're genetically different, but it's understood how they are different. And these are not outbred mice, these are mice which are generated not by us, this is a whole consortium that creates these mice. These are mice that were originally generated from two parents, a mother and a father, from two well-known laboratory strains, C57 Black 6 and DBA. These two parents were crossed, and then through some additional crosses and a lot of inbreeding later on, which I don't want to dwell on in detail, about 180 strains were generated. So each mouse in each strain is the same, is genetically identical to the other mouse in the same strain. But the mice across strains are genetically different, and they're different approximately like brothers and sisters in a family, because they come from the same parent here. So basically the alleles from these two parents were shuffled around and create 180 combinations of alleles. We know that in every locus there's either the Black 6 or the DBA allele, but the distribution is different. So we basically create a gigantic family of mice, of which we have from each brother and sister, we have multiple numbers of individuals which are genetically identical, but across they're different. So this is a resource that we can use, the known genomic variability, to relate this to phenotypes, and this consortium, which is called BXD consortium, Black 6 times DBA, measured about 300 phenotypes on these mice. This is an amazing resource because we can relate molecular patterns we measure in these mice to these phenotypes. Just to indicate how interesting this is. This is two phenotypes which are measured in each one of these strains, and the values are listed here. So we see here one of these phenotypes is VO2 max. So this is for athletes are interested in this phenotype because it basically means how much oxygen your body is able to burn when you're an athlete, when you're running or driving with a bicycle or whatever. And we can see here, this is the Black 6, and this is the DBA strain and here, this is on normal food, and this is on high-fat diet. What we see here is that these two strains inherently have roughly similar VO2 max when they are lean, eat normal food. When they have fat food and get fat, they both drop their VO2 max, and they drop it to a different extent. This is not super exciting, but it is remarkable that we can see this and they behave differently here. There's another phenotype which I chose here as an example. Serum alkaline phosphatase actually used as a clinical marker for bone metabolism, and we see that in this case the two founder strains are quite different, but when they are on high fat diet, they have the same value. So basically, we see here that these phenotypes, which are measurable, are different across the parents, and the parents also react differently to a perturbation, a metabolic perturbation, these phenotypes. Now, this is not super remarkable, it becomes phenomenally interesting when you look at the strains. So here is the 180 strains, and this would be the VO2 max phenotype. We see here that these two parents have very high VO2 max, but most of their offspring have much lower VO2 max. If you remember anything about Mendelian genetics, this is not explainable by Mendelian genetics. You would assume in Mendelian genetics, if there's a marker, an allele that creates this phenotype, you would have either the offspring would have either the one from the mother, the one from the father, or something in between. And here, most of the phenotypes are outside of the range defined by the parents. And this is the same for the other phenotype as well. Serum alkaline phosphatase is the same story. So what this immediately points to, that by reducing these phenotypes to a specific gene or a specific locus will not work. It's a much more complicated arrangement how these phenotypes arise, and I think it's probably true for most phenotypes also in disease. So we wanted to see whether we can use molecular level measurements, learn something, and do this presumably reproducibly across these strains, and then relate these phenotypes relate these molecular patterns to a phenotype, to learn mechanistically, or at least first by association, eventually mechanistically, how the genomic variants caused a phenotype. So this is the intent of this short study, or study I'm briefly summarizing here. This was work from a postdoc, Evan Williams, in the lab, and another postdoc, Yibo Wu. They took a number of these strains, about 40 strains, in both normal food or in high-fat diet, and then they extracted livers from these mice and quantified a number of proteins. They used SRM, later on they used DIA. I'm showing some results for SRM, but basically the same results, but of course with more proteins were measured by DIA. We also measured transcriptomic measurements. So we had the genomic background, transcript measurements, and proteome measurements. For the proteome measurements, so I think there's a slide missing. What I want to show is just the results. But basically, these results are 40 strains times 2 metabolic states times about 190 proteins that were quantified with very good reproducibility. I think it's coming later on. And then so we can now relate the protein abundance. So we have now basically protein abundance measured over a number of mice. And we can relate this by association to basically correlate the abundance of proteins to the presence of a locus, of a particular allele in a particular locus. And this is referred to as quantitative trait, locus analysis. This is the spread, this is the summary sheet of the data, and this is what the results show. We can relate some of the variability, basically whether it's a high level of an enzyme or a protein, or a low level, we can relate this to a genomic variant, because we know for each locus which genomic variant is present, either the one from the mother or the one from the father. So this is referred to as quantitative trait locus mapping, or QTL mapping. It establishes a relationship between a genetic locus that determines the abundance of, in this case, a protein, or if it is done at the level of transcripts, between the genetic locus and the abundance of a transcript. So we identified among these 190 or so proteins measured, 44 such QTLs, 45 the lowest side that affect the mechanistically, we don't know how, but they affect the abundance of specific proteins that were measured in across these mice. So we basically use the fact that these mice are variable, and we use the whole data set to establish this association between genomic locus and the abundance of a protein. Okay, so now this is done quite a lot, but when it became interesting, when we tried to use these QTLs and to see whether we could learn something biological, we now like to translate what is simply a statistical association, which also happens, for instance, in GWAS studies a lot, to try to go further and try to relate this to a mechanism, using the richness of the data created from these variable mouse strains. So one of these QTLs, just as an example, mapped to an enzyme that is the last step here of this pathway, biosynthetic or biodegrading pathway, where branched chain amino acids like lysine are degraded. So this is a very well-known biochemical pathway. People know the enzymes that carry out the steps, the stepwise process, and these enzymes are known, and the products, the intermediate products they produce are also known. So basically we have an enzyme down here that at the end of this degradation pathway, and we have a locus that determines whether there is a lot of this enzyme or a small amount of this enzyme present in the cell. Genetic determinant of abundance is QTL. And so now we can measure also by metabolic measurements these intermediate products. And so we can correlate these intermediate products, for instance, this amino adipate, which is simply a degradation product of this branched chain amino acid, and we can correlate it with the abundance of the enzyme. And we see that there's an inverse correlation, and that the two intermediate products here correlate positively with each other. So what does this mean? It basically means that we can translate this into a mechanism where we say we have a pathway here, and then when this pathway is restricted at the bottom, because the last enzyme in this pathway is not present at a high abundance, the flux through this is low, and of course the other enzymes are all higher, and these intermediate products pile up. So we have high levels of intermediate metabolites if there is a low level of the enzyme present. And conversely, if there's a high level of the enzyme present, then the flux is high and the intermediate products don't accumulate. So this is showing basically that we have a genetic control, a locus that controls the abundance of an enzyme, and that regulates the abundance of intermediate metabolites. And now this is becoming interesting when we then looked in the literature and found that this amino adipic acid, is a biomarker for diabetes risk. So this is a paper that was published not by us. This is from the literature. Actually, Bob Gersten, who is here in Boston, very prominent researcher. And so they established from the Framingham study, which is a large population-based study, they established that this particular intermediate metabolite is an indicator for diabetes risk. So now we can again say, if we put this in perspective of this QTL study, we can say we have identified a genomic locus which determines the abundance of a specific enzyme, and if this enzyme is low, this compound accumulates and is considered, based on epidemiological study, a biomarker for diabetes. So it links basically a molecule that's simply been found by association in epidemiology. It links it to a genomic variant in the mouse, and this was later confirmed this also happens in humans. So this is a way to go from associations, initially QTL mapping and then also epidemiology, to define kind of a mechanistically plausible way how this actually might work. And this is only possible, this would not be possible, of course, if we did a controlled experiment where we would take a single strain of a mouse and manipulate this mouse in some way and keep everything else controlled. So we try to take advantage of the variability of this population, this BXD population, to generate data sets where we can learn something from basically the introduced perturbations, genomic perturbations that have been established through evolution. So this was an example where we tried to say genomic variability is not a threat, but where we say we can use it to our advantage to learn some new biology which would not be, or would be very hard to find out if we used a single strain and kept every variable constant. Okay, so this was kind of justification for this problem. I tried to first address the kind of societal political reasons, and now also the scientific reasons. Of course, we can only do such studies if we can generate reproducible data and analyze the data reproducibly. Okay, now I would like to get to the point on this nature table, here, the nature menu, and ask the question, given the techniques we learned and discussed here, if two or more people have the same proteomic data, can they generate the same results? This is at the core of what you learned now in these 2 weeks. And so there was a community-based study in the DIA field. This has actually not been done in DDA mass spectrometry because it's very hard to actually do. But in DIA, this was possible because of the inherent properties of the technique. So we generated in our group a data set where 3 proteomes were mixed together at certain ratios in 2 samples. So here would be 20% E. coli, 15% yeast, 65% human proteins. And then in the second sample would be 5% E. coli, 30% yeast, and 65% human proteins. So if we compare these 2 samples with each other, every E. coli protein should have a ratio of 1 to 4 for the E. coli and 1 to 2 for the yeast proteins. And then we would have an accurate outcome, basically establish a ground truth of the ratios of the proteins from a particular species that should be observed in the analysis of the data. These samples were acquired by DIA swath mass spectrometry in 2 mass spectrometers, and then everyone who at the time had created a software tool to analyze this DIA data got together. And at the time this was 5 software tools: OpenSwath, Swath 2.0, Skyline, Spectronaut from a private company, and Umpire, which is working slightly differently. I don't know whether you worked with the DIA Umpire. It's a library free. I mean, it creates libraries on the fly, but it otherwise uses the same analysis principles. And everyone analyzed this data, the same kind of ground truth data sets, and then we saw what came out. So this is the experimental design, which I already explained. And here's now some results. I don't want to go through all these results. I mean, this is a lot of results and a lot of things we could discuss. I just want to focus on some of the key points. The first key point was that when everybody from these 5 developers run their tool over the data, they discovered room for improvement. Let's put it this way. So the first iteration, when we compared the first iteration results to the ground truth and between software tools, there clearly was room for improvement. Then everyone got together and this was discussed, and everyone improved or worked on their software tool, and then the results improved. So this would be the different software tools. This would be the protein level data. So we see the coli, the human, and the this would be the yeast, the human, the coli. It's 4-fold difference, 2-fold difference. And we see that for the most part, the measured ratios are actually quite accurate. And we see, of course, for low intensity signals, which are on the left side here, that the spread becomes bigger. This is typical for any analytical technique. When you get closer signal to noise, the variability goes up. So we could say that from the point of view of quantification, determining abundance ratios, every one of these tools, after an iterative improvement, produced fairly accurate and quite similar results to each other. This is a good thing for the field, because we know, we can, I think, state that if someone creates DIA data and uses any one of these tools, they will be likely, if they apply the tools properly, they will likely create quantitatively accurate, in terms of ratios, results, irrespective of which tool they they use. Now, in the meantime, there's been a whole slew of additional tools that have been developed, which have not been actually benchmarked. It would be maybe a good thing for everyone who has a new tool to also run this against this ground truth data set and see what happens. Pedro Navarro, who was Pedro Navarro here, the first author who actually did this analysis, he created kind of a comparison workbench. So if someone is interested to do something like that, you could, in fact, download all these programs that now exist, download the data, and then use this workbench to do this comparison. It would actually be an interesting service to the community. Okay, so this was about quantification, but we're also, of course, interested in identification of proteins, and this is typical kind of Venn diagrams. I just want to point out that about close to 5,000 proteins were identified, most of them by old tools, or by two tools, by all tools minus one. And there is relatively few proteins, only single-digit proteins, that were discovered by only one of the tools. So we can also state here that if these samples are analyzed with any one of these 5 tools that were tested, that the identifications that came out are very consistent, independent of the software tool used. This is also a good thing for reproducibility issues, but it also precludes, I think, endless cycles of students going and try to optimize their search tool. And we have experienced this ourselves in the field of mass spectrometry, that when someone comes up with a new search tool, for instance, in DDA mass spectrometry, there must be now dozens of search tools. Everyone writes a paper and says, my tool is better, it sees more proteins than all the other tools before. And then inevitably someone from the lab goes and says, I want to test that. And then 2 months later they come and say, well, I'm not so sure whether it is actually better. Also, there's an enormous amount of energy and I think time is wasted in comparisons, which I think here are no longer really necessary if you use one of these tools and use it properly. So I will conclude here that from this question, if 2 or more people have the same proteomic data, can they generate the same results? I would say the answer in the AA world is yes, and we could then conclude that the variability we may observe in DIA mass spectrometry, that this is not coming from the software tools, which is very different from the old times some years ago in proteomics. I cited on Wednesday a study from Bell, which is a very simple study, 20 proteins that were equimolar, could not be identified consistently across laboratories. So there's been enormous progress in the field through the tools that, and I would say for this question we can give a positive answer. Now let's go to the next point here. This would be data acquisition. We also wanted to ask, this is one of the points that Nature also has there on the menu. And again, we want to undertake a community search or community-based assessment, whether 2 or more laboratories that have the same sample and run it through a machine, mass spectrometer, whether they can come up with results that are reproducible and similar to each other. So this is the design of the study. We generated a lysate of HEC293 cells, a commonly used human cell line, and then we spiked into this lysate 30 isotope labeled peptides at various ratios, so at like 3-fold dilution steps. And we generated a range of samples that would cover collectively several, I think 5 orders of magnitude of dynamic range, and in each of these samples we would be able to basically measure dilution series of these isotope labeled peptides if the samples were run in a particular sequence. So this was to address the question whether we can identify proteins consistently from the sample, which is a typical complex sample as is experienced in mass spectrometry today, and whether we would get reproducible dilution curves, basically a reproducible quantification. These samples were sent to 11 groups that participated from various countries and continents, and then everyone agreed to run these samples in a scheme that we predetermined on day 1, let's say on a Monday, Wednesday, and Friday, and they would run on Monday the samples in a particular sequence, which would allow us to then reconstruct these isotope dilution series. So everyone committed to running 21 samples and then sent the data back, and we answered the questions which are posed here. Do we detect a consistent set of proteins if the same sample is acquired in different instruments? And what is the quantitative characteristics across labs? So this is what came out. I already showed this last Wednesday, and now I want to go into a little bit more detail. So the first thing, so this is what we observed when we looked at each run, basically individually. So each file was searched against the library, and the number of proteins were identified. We see that when you analyze these samples, they are quite consistent, which is a good thing. We also learned, and that this is now just a mock-up, but we learned that when we keep adding data, so when we are fairly confident from the underlying statistical model, as I'm sure this was discussed in the course, that there is issues of false positives, control of FDR, and so we feel fairly confident that we could control the FDR in a particular sample. But what happened when we added 100 or 200 or more samples as an aggregate? Because we have basically a large number of samples, and then we observed that the more samples we added, the more proteins seemingly got identified. So this is a surefire indication that there is an accumulation of false positives. And I think this should be always done. I don't know to what extent this was discussed. I guess this was discussed in the course, that if one has a large number of replicates, one should always do some kind of accumulation plot, and if the curve keeps creeping up, this is a very bad sign. This basically means almost in all cases that the control of the false positive rates is not under control. I'm going to illustrate this with an example. So this is from the data-dependent analysis mass spectrometry, and this was eventually translated, these insights were translated in a tool by a student at the time in our group, Manfred Klassen, and he analyzed this accumulation of false positives, which we first realized when we started to collect data from multiple labs and try to build a composite picture of the human proteome. And what we observed, so even if we controlled what typically is done, the FDR of peptide identification, let's say at 1% or at 0.1%, it doesn't really matter the value, the principle remains the same. We would see that at the true positive identifications are seen over and over and over again, because they're true, and we know then that, so this would be these ones here, they would concentrate on a relatively, well, on a set of proteins which are certainly identified in the sample. If we did this again, and if we did 100 times, the true proteins would still be identified. We simply get a large number of repeat observations of these true positive proteins. But for the negative ones, the false positives, which may be 1%, maybe 0.1%, or whatever we choose as a threshold, for them there's no reason why they would associate with any particular protein. They have the same likelihood to be matched to any conceivable protein in the proteome. So if you do this long enough, you would seemingly have evidence that you have identified every protein. And so then we would say these proteins here are certainly truly identified. Those here are basically seemingly or artifactually identified because they are an accumulation. They only have hits by false positives. Of course, we don't know a priori what is a true positive and a false positive. And Manfred Klassen then worked an algorithm out which differentiated these, which basically identified those proteins which he believes are truly identified in very large data sets. And the curves we should see when we have increased the number of basically data points from low number to very high number, we should see something like that. Initially, the number of identification protein or peptide discoveries goes up, and then with proper control of FDR, it should flatten out here. And if it kind of keeps creeping up here, this delta is almost certainly an accumulation of false positives. So this was in the field of DDA was solved. In the field of DIA, this is now also resolved through a community effort again, where a number of labs, including Brendan and Mike McCoss and Alexei Nishvisky, got together and said we need to address this issue for DIA to also get, when we increase the number of replicates, that we come up with a curve that saturates out and is then stable. We don't accumulate false positives. So this was a study that was done by by a student, or headed by a student in our group, but you see here also Alexis there, and you see also somewhere Mike McCoss. Here is Mike as a co-author, and Brendan. Again, this community came nicely together to solve a problem that was found to be found empirically from the first very large data sets that were acquired. Okay, so I think I skip this. I come to the quantification. Here now we now said that the overall protein identification rate, or the number of proteins and the type of proteins identified, was quite consistent. So we can say, from the identification point of view, the method is reproducible across laboratories. And now we wanted to talk about quantification. These are the dilution series of some of these peptides in the sample, and each color here is a different laboratory of the 11. We see that they have fairly parallel curves, which is good, but they're slightly offset. So now we can simply re-normalize these curves to each other by doing some batch correction, because in one way the instrument was slightly lower sensitivity because it was dirty, and then with some very simple normalization, we see that these dilution curves of aqua peptides, isotope level peptides, that were spiked into these samples very nicely converge between the laboratories. So this is very encouraging over several orders of magnitude. So this is very encouraging because we can say that if samples are analyzed, two or more samples are analyzed, or identical samples are analyzed in different laboratories, that the results are quite consistent both from point of view of identification of proteins, but also from the point of view of quantification, which is fairly rigorous with these isotope level peptides. And of course they were embedded in this complex matrix of hex cells, and so this is encouraging from the mass spectrometric side. So we have asked so far two questions. If two or more people have the same proteomic results or data, can they generate the same results? And we said we confidently can say this is true if they use any of these benchmark tools. We also asked if two or more labs or two more people have the same sample, can they generate the same proteomic results? And the answer here is it's not the same, it's not identical, but it is actually quite highly reproducible, both from the level of quantification, but also from the level of identification. The only kind of requirements that were sent to these laboratories which run these samples was to say they should use all the same mass spectrometer. We don't know how different mass spectrometers behave across different types and brands, but this was all with SciEx instruments, because at the time this was the only real instrument that could really do this type of acquisition. And I would be certainly surprised if one did the same thing on a Thermo instrument or a timsTOF, if the results were very different. In fact, Thermo has done now repeated basically the same design of study, and they are, I think at ASMS they want to announce how this worked, and the rumors are it worked also very nicely. So I think we can say that from the point of view of reproducibility of research results, that if we ask these two questions, we can in proteomics quite confidently say we are in quite good shape. Of course, it's not perfect, but we are in pretty good shape. I would like to come to the last point here, and that is reagents, cells, tissues, and so on. Basically, the basis for the experiment. And we, again, undertook a study which has to do with HeLa cells. These are cultured cells, which are cancer cells, which are very often used in clinical or basic research. So this is simply to show that these are very widely used cells. There is, at the time, there were 97, when we made these slides, there were 97,000 publications which used HeLa cells for their studies. Now it is more than 100,000. And so this is a sizable effort. And we wanted to ask now here that if different people used HeLa cells, well, HeLa cells in different laboratories, and they did the same experiment, whether they would get the same research results. And that is, so the reason to ask that is not a trivial matter, because these are cells which have been in culture for a long time, several decades, and it is plausible that they diverged, because they initially were cancer cells, and many cancer cells have unstable chromosomes. And there's also another issue, that is the overgrowth of cells. So people might work with other cells, like HEK cells. All of a sudden, they actually grow HeLa cells. This is not what we address. We address the question, if two people do the same experiment in genuine HeLa cells, do they get the same results? Okay, so this was the study design. We collected, and this was work from Yansheng Lu, who was a postdoc. This actually was just published a couple of months ago. And he collected HeLa cells from different laboratories that we wrote letters to in different research institutions. And they have numbers, so they'll be collected like 15 such cells. They're ostensibly all the same. People would write in the paper, I did an experiment with HeLa. And there's two big groups. One is called Kyoto variant and CCL2, which is another branch. And so these were, usually this is not even specified in the paper. And we were lucky that one collaborator, the group of Wolf Hart, a microbiologist in ETH, he grew cells for a long time and periodically froze them down. So every 2 or 3 months he froze them down, so we also had a time axis to see how stable they were if they're grown in the same lab under the same conditions. So this is the experimental setup, or the source of the material, and then Yansheng took each one of the cell lines, grew them in the laboratory under the same medium, same conditions, same time, and then he measured did a number of measurements. He measured copy number variants in the basically genomic analysis. He measured also mutations, which I'm not talking about. He measured the RNA profiles, steady-state proteome, and he measured the protein turnover. And then we measured also the response of the respective proteome to perturbation. So these are several layers of data which he created from each one of these cells, and then we could learn something about the question: are they different? Do they react differently? Or are they all the same, and do they give the same result if we do an experiment? First observation was that they are quite different. Simply based on the molecular makeup, this would be a kind of a comparison between the cells, and we see here they're spread out in this two-dimensional graph, and they're probably more spread out than some of the cells, which are, these would be lung cells, and they're cells from different tissue origins. They tend to be relatively closely clustered together, but these cells are actually quite far apart, even though they originally were from the same person long time ago. So this already shows that they are somewhat different. And then we measured, or Yansheng measured, a very simple phenotype, this doubling rate, and this would be the cell lines, and this would be the hours they take to divide, and this goes from some like 18 hours or so to more than 30 hours, maybe 36 hours, 32 hours or so. So we see that under the same conditions, the ability of the cells to grow has diverged quite substantially. So these are simply two very crude phenotypes to show that we have evidence to believe that the cells have diverged. Now we went to the molecular levels. Skip this and show that we are able to detect differences. This would be so he quantified all these proteins by spot mass spectrometry, and they quantified about 5,000 proteins. And when he did repeat analysis, whole process replicates, he has a correlation coefficient higher than when he does between cells, and he has higher correlation than when he does between the two branches, CCL2 and Kyoto. So this is what you would expect, that the technical variability is smaller than the biological reproducibility between same strain names, the two branches, or between the branches. So this is a plausible result to show that we can detect credible differences within the confines of the technique. Okay, now I show some results what came out. This is the genotypic variability between these cell lines at the level of copy numbers. So we see here the chromosomes aligned, chromosome 1, 2, 3, and so on. and the cell lines are the concentric rings. So this would be one of the cell lines with these chromosomes, and the second ring the same for the next cell line. And we see here that obviously right away we see very distinctive patterns. Green means low copy number, and red means amplification of this particular loci through copy number variation. And we see differences which are between cells. We see differences which are quite distinctive, and we see big blocks. And here, for instance, is a block that's consistently multiplied, and we also see blocks which are multiplied only in some of the cells but not in others. So they are genomically quite different, and that was observed. Interestingly, when we compare these two the outer cell lines here, 12 and 14, these were cells which were frozen down from the same lab, Wolf Hart's lab, within about 3 months. So they already show, within 3 months of experimentation in the same laboratory, show substantial differences in their copy number situation. We see a very distinctive difference here, in that the one cell has low copy number here, and the other one has high copy number, basically for all of this chromosome. And there's other differences as well between these two cells. So that means that if someone is doing, a student is doing a study, let's say over the duration of a thesis, and they start to do an experiment in the first month, establish the whole system, and do the experiment, and then want to repeat it, maybe to be sure when they publish a paper, they may not be doing the same experiment in the sense that the cells have evolved. So now we could say, well, that's just the way it is, but maybe these copy numbers are buffered, and it doesn't have any effect on the phenotype or the expressed information. That's not so. So we now show here for 2 chromosomes only the abundance, how these copy number variants are translated into transcripts and into proteins. And when we first saw this, I was very fascinated by these patterns, because we see really interesting, we make really interesting observations. So for instance, we see here that this whole block of chromosomes here, of chromosome 1, is essentially green, has very low copy number, maybe 2. And we see then at the transcript level, these very distinctive patterns, red and green here at the genomic level, are largely washed out. And they're even more washed out at the protein level. So what this means is that you see increases in transcripts and increases in proteins for regions where the corresponding RNA, the corresponding gene, is not really multiplied. So this points again to very complicated, complex regulatory mechanisms, that how the cell interprets an increase of copy numbers in one particular locus. So it is not so that just this locus is then, if there are 4 times as many copies per cell, that you generate 4 times as many transcripts and 4 times as many proteins. The whole thing is obviously much more complicated. So this is, I think, fascinating to see, but it's just graphical. We then try to figure out whether we could derive some rules, like which proteins might react more directly to a copy number increase, and which ones might be buffered down, or we try to learn some rules. And to do that, Yancheng did some correlation analysis, where he basically correlated the copy number variance with mRNA level. So we see here is a broad distribution, so it's clearly not strongly correlated. When there's more copy numbers, it doesn't mean that automatically more of this transcript is being generated. So for each one of these correlations, copy number mRNA, RNA, protein, CM, copy number, protein, the correlation is there, but it is relatively low. So this is again descriptive, basically a numerical description of the graph before, and now we try to figure out whether we can derive some rules. And one of the observations, or one of the hypotheses we tested, or Yancheng tested, was to say that a lot of the buffering that occurs, so let's say the copy numbers go up, but the protein does not go up. might happen at the level of the organization of proteins in complexes. So this seems to be the case. So I've shown now this, now to show this, explain this graph. So when we correlate copy numbers with mRNA, so I should say first, we distinguish now here, we use these correlations between copy number variation in mRNA and copy number in protein, and we segregate this into two groups. The one group is genes that are known to be in protein complexes, and the other is genes which are not known to be in complex. It doesn't mean they're not in a complex, but there's no evidence for it. So we then ask whether the complex in proteins or the corresponding RNA and the complex out, so those where there's no evidence for being in a complex, whether those are the blue ones, these are the reddish ones, whether they have different behavior in terms of this correlation analysis. If that was the case, we would say that the fact that they're in a complex has an effect on the on these proteins, how they respond to a higher copy number. Okay, so this is what we see here at the RNA level. If we correlate copy number to RNA, there is no difference detectable whether or not the protein, whether the RNA that eventually leads to protein, whether the protein is in or out of a complex. There's no difference. When we look at the proteins here, we see a difference. We see that the complex out proteins are more strongly correlating than the complex in proteins. That means the complex in proteins are less strongly following the copy number variation, and we will call this they're buffered down. And they're buffered down because when we correlate the copy number variant to the degradation rate, they're also dysregulated, or they're also diverged in a different way. So basically what we conclude here from this very cursory analysis, based on a lot of data, is that proteins that are being synthesized because there's more higher copy numbers present in the cell, that these proteins are translated equally into transcripts or into transcript, whether the eventual protein is going to be in or out of a complex. But when the protein is made through translation, if the protein is going to be in a complex, it is down buffered through degradation because it finds no way to go. If the other components of this complex are not also upregulated. Basically the cell will, let's imagine we have a complex of 4 proteins. Protein 1, 2, 3 would be synthesized at a specific level, and then through this copy number, the cell produces now double the protein amount as normally foreseen, concordant with the stoichiometry, but the excess protein does not have a place to go and is degraded and basically moved away. So what we can say is that what we learn here is that this, at least one factor, is certainly not the only factor, of this very complicated way how genomic variants are translated into transcripts and proteins. At least one factor is that they're buffered, the abundance of proteins is buffered at the level of the organization of the proteins into modules. This has, of course, implications for everyone who is in the clinical arena doing, for instance, RNA analysis. They might find between cancer cells and the control tissue upregulation of certain transcripts. They might identify these very confidently, but it may not matter because the protein may be downregulated. In fact, there's many cases like that known. So I'd like to now, I think I skipped this for the interest of time. This was simply to show that these cells were not only had a different molecular makeup, but they also reacted differently to a phenotypic measurement, which is invasion of the cells by a bacterium. So this is just one of many phenotypes we could measure, but so what we could say is the cells, they were collected as ostensibly the same cells. They were collected from different laboratories. We show that they are actually different cells, even though they have the same name, from their molecular makeup. And they also do react differently if you do something to these cells. So that basically means that if two people are doing the same experiment in HeLa cells, they will actually not do the same experiment unless they're extremely careful and really document what these cells are at the moment. And if they want to repeat this same experiment, they would have to go back and thaw up a vial of cells, which goes back to the same root, and then grow them for a similar number of, preferably low number of passages, and then repeat the experiment. So simply growing the cells for a year and then do it again will inevitably mean they're already doing a different experiment. So this has, I think, implications for experimental biology publications, and that's of course not just true for HeLa cells. This is true for many other cancer cell lines which are genetically unstable. And so when I started out, to finish off now or summarize, I started out by framing this presentation in the context of reproducibility of research results. I tried to first explain why this is important for, let's say, political reasons, but also that if we work in biology, this is different from physics or chemistry. We have an inherently underlying biological diversity that is inherent in biological systems. And this is certainly true in outbred populations. So we can say this is a problem, or we can say this is an opportunity. And I tried to show an example where we use this inherent genomic variability as an opportunity to learn new biology by using the breadth of data that is collectible now by mass spectrometry, of course also by other high throughput techniques. Then I tried to pose 3 questions and say, where in the field of proteomics, when we have same data, can we get same results? If you have same samples, can we get same results? And if we do the same experiment, do we get same results? And I would say in the technical arena, where we do a measurement, we have tools that try to get results like protein quantities and identities from this result, from this data. We are doing quite well in proteomics. It is quite well benchmarked in terms of the acquisition of data, in terms of the analysis of data. So this is encouraging, but where we see clearly a big source of reproducibility problem is the biological basis of the material that we use, in this case cell lines. And this will incidentally be similar to mice or fish or worms, which also evolve, of course not at the same rate, but there's many strains that are being used and known, and if one is not careful and documents what strain an experiment was done in, then a researcher might get quite substantially different results. So I would say that if we are confronted, just as a last statement, I would say that if we are confronted as life scientists with this reproducibility crisis, we should not be defensive. We should say, look, there is an issue of reproducibility, but it is, in most cases, I'm not saying no one cheats, I'm not saying no one is incompetent, but most people don't cheat and most people are not incompetent. And I would say this problem of reproducibility is inherent in the biological matter, and I think this is a very inherently positive statement. We need, of course, learn how to control that, or to at least document this variability, that we could say, if you use exactly these cells which have the same genetic makeup, you can actually generate the same results. But if the underlying material is not documented, of course the results will be different and appear irreproducible, even though no one made a mistake and no one tried to cheat. So with that, I would like to finish. I'd like to acknowledge some of the members of the lab whose data I showed here. So this technique, the DIA technique, and many applications of it were done by initially Ludovic Schier and Pedro Navarro, together with AB Science. This is now, of course, a broad field with different instrument manufacturers supporting this technology, which is great. I talked about the software comparison. Cross-lab software benchmarking was carried out by essentially Pedro Navarro with contributions and also donation of everyone listed here who donated their software and got together and did this optimization of software tools. And the cross-lab study was done by Ben Collins and Christy Hunter. And this HeLa study I showed last was from Yansheng Lu, who has now his own lab. And we're particularly grateful to Wolf Hart, who gave us access to cells we froze down every couple of months, which was kind of nice chronological track. So I would like to thank you and to answer questions. And if there are no questions, I would wish you a safe return. And I hope you learned a lot in these 2 weeks or in this course. Thank you very much.
With work, you showed the HeLa work. You showed the correlation between genomic copy transcripts, mRNA transcripts, and proteins. These are the amount of these biological materials. Is it possible to correlate these three with functions?
Yes, to some extent. Of course, you have to have a function you want to correlate with. And so the part I skipped for a reason of time was the part where we actually used one particular function, and it's somewhat convoluted assay, but these cells, so there is the observation from this microbiologist Wolf Hart that cells get invaded by Salmonella, and that he found in a screen, he found a microRNA that interrupted that. So he basically made them, protected them from being invaded. And so we used this the observation he had made in his lab, and we asked if we subject these cells to this particular microRNA, do they react similarly to prevent this invasion, or do they react differently? And it turns out they react differently. They generated, if you put this microRNA on these cells, they show a different response pattern at the protein level, and then this correlated. Some proteins of this response pattern correlated whether the cells still allowed the bacterium to invade or not. So this was just one example, of course, because we had access to this particular assay where we could clearly show that the molecular makeup and the response of these cells to a perturbation, this LED7 microRNA, correlated and probably mechanistically is rooted in the ability of the bacteria to go into these cells. And so that's the beauty of this BXD mouse system. We have hundreds of phenotypes that you can now, if you have molecular patterns, you can correlate with any of these phenotypes. And I think this is a great resource. Generally now I think we are much better in measuring molecular patterns than measuring phenotypes on cohorts. Yes?
So you talked a lot about reproducibility and sort of trying to, when you show the beautiful graph when people say, yeah, there's no reproducibility in science. Do you think, in a broader spectrum, could we generate some sort of standard or a reference mark, as Michael McCoss talked about, there's not a true reference, but like a human reference to show like the copy number variations. I think HeLa was pretty consistent, and it changed mRNA and transfer, but can we sort of, within our experiment, if we're studying HeLa or mouse, generate a standard that when we normalize our experiment across labs, then we can find that there is reproducibility, just there's not a reference for us to look in.
Yeah, it's just, I think this is a very good idea. I mean, something like that probably should happen. I think the first state, or the first level, which is the least intrusive, would be simply to document better what that you would, maybe if someone is doing an experiment with HeLa cells or whatever cell line, that they would say, I take a little aliquot and I go and measure a transcript profile, which costs probably now, I don't know what this costs, $500 or something, and then say, I append this to this cells or something like that. But this would be already much more elaborate, that you have some kind of reference. But of course, if you have a universal reference and you observe a difference, you simply know it's different, but it will be very hard to say from the difference you observe in your study how this translates into some mechanism, because I think this is very, very hard to figure out, but I think something along these lines will happen. So now we in a month, EMBO agreed to sponsor, EMBO is the European Molecular Biology Organization, agreed to sponsor a workshop where we want to discuss these issues and say what are the implications for pharma researchers. They do many pharma companies do large-scale drug screens on HeLa cells or other cancer cell lines. What is the implications for FDA filing, for portability of results to other cell lines, and so on, and also for basic science. So the outcome should be some kind of conclusion how scientists who do experiments with these cell lines would report in a paper what they were actually doing. This might be that they simply describe the number of passages since the original file was thought up, that they might do some proteome map, which would be very easy to do, or whatever. But we will see what comes out. I think it's great that we have. It's about 25 people will be there from various branches, from publishing houses, from granting agencies, from pharma, from basic science, to discuss what does it actually mean. For that, hopefully, we could then say, well, there is a reproducibility issue. It is inherent in biological system. That's how we can at least in part address it. Yes.
Can we take this data and reanalyze them from a different perspective? I don't know if you can analyze the post-translational modification PTM like ubiquitin and the proteasome pathway, especially in the cases where you argue about more degradation, correlate them with the passage number for the cell line.
Yes, so this data is all accessible. If you wanted to research it, since it is DAA data, you would simply need, if you for particular modification, you would need to generate some form of a library, and then you could certainly do that. Of course, it's not fractionated, it's not enriched.
For
yeah.
PTM. Yeah. But you could still see some,
yeah, you could do that.
So you know the passage numbers for each of these cells?
As far as the labs that donate the cells knew. Sometimes they didn't know. They just said, well, student Peter got it from Miller's lab.
No, but the turnover number, because you measure a lot of proteome in the different stages. I'm just curious, in the turnover type of experiment for the proteome, was any difference, huge difference between one type of passage, early passages, or you didn't enter in this detail?
So we, as far as the number of passages is known, we could do that, and the Ansheng did this for these two cell lines where we have documented linear track over a number of cycles. But generally, I think it's simply not possible to obtain the passages because people don't know. It's not recorded. Cells sometimes reside for years in the same lab, and one person gives them to the other, and because they're so easy to grow. But if you're interested, it would be great if you did something with the data. It's all accessible as far as we could reconstruct the description. Yes?
How would you, given the speed of advancements that have happened in the field, particularly in the last decade, to someone that's particularly not well-versed in our field, how would you address any skepticism from them if we were having to justify the overhead things for large consumer purchases like this? We're just now getting to this kind of level. It's something I've encountered probably
Specifically for proteomics, or
specifically for proteomics.
Yes. So I think this is a good point. Proteomics has in many areas, also with funding agencies, a bad reputation. We had a number of crises which gave it a bad taste. So there was notable this cell-Toff crisis, where a peak was developed which made claims which could not be fulfilled. And then, but since then, what has happened? But there's still the lingering. There's basically two comments we get to your in the context of your question. One is it doesn't work. Proteomics doesn't work, and that I think can now be addressed with publication. That's why we all spend considerable effort with a lot of colleagues to try to do this community study. You can say, look, it actually does work, and it does produce with limits. And no one says it perfectly. With limits, it produces high quality. And the second objection usually is, well, it works, but only in a few highly specialized labs. And this is also no longer the case. I think especially with these EAA methods, also to some extent with these like labeling, isobaric labeling techniques, which are, I think, more complicated to use because you have to do some chemistry. But they also have clearly been shown to produce very high quality data if it's done properly. I think it is harder to do with labeling than without labeling, but it is also certainly possible. So I would say, well, I mean, this is best argument would probably be to say, look, it works now, we can do it, and if you don't buy me the machine, my institution will be left. That's how many react to. I mean, that's why everyone buys immediately if Illumina comes out with some kind of new sequencing instrument. Everyone immediately will buy one if they can, because they say, well, otherwise we fear of being left behind. I think what we have in proteomics not managed to create is the impression that if you don't do it and if you don't buy the machines, that you're going to be left behind. And I think this is actually probably increasingly a